Welcome to today's TRI Research Translation Committee seminar on innovative clinical trial design for investigator-led trials. So before we start, I'd like to start by acknowledging the traditional custodians of the land on which we are today, the Turrbal and Jagera people, and we recognize their continuing connection to land, waters, and community. We pay deep respect to them and their cultures, to the elders past and present and emerging. So today we'll be hearing about the establishment of Clinical Trials Centre at the University of Queensland and the new capability for innovation in clinical trials and what it means for TRI researchers. You'll also hear about the innovative clinical design journey and a vision for success from Professor Nadine Foster, Professor Andrew Martin, and Associate Professor Ingrid Hickman. So in late 2022, UQ launched the Vice Chancellor's Health Research Accelerator, or the HERA initiative. So through this initiative, UQ is investing in health and medical research and capabilities to address the most pressing health and medical research challenges of our time. So one of the 7 new HERA initiatives is the ULTRA, or the University of Queensland's Clinical Trials Capability. The ULTRA program has been created by the university to build capability and innovative clinical trial design, conduct analysis, and translation. Its vision for ULTRA is to create a world-leading interdisciplinary team focused on innovation in clinical trials that leads to more rapid results and improved patient outcomes. So today's talks will be followed by a Q&A session. So for those in audience, please save your questions for then. And for those online, please feel free to send through your questions and chat via the Zoom function. So Amelia will be next to introduce the speakers. Thanks.
Thank you, Amila. So I have the pleasure of welcoming our first speaker today, Professor Nadine Foster. Professor Foster is the Academic Director of UQ Clinical Trials Centre and lead for the ULTRA initiative, and Director of the STARS Education and Research Alliance, which is part of the University of Queensland and Metro North Health. Professor Foster is an NHMRC Leadership Fellow and an internationally recognised expert in musculoskeletal rehabilitation health services research and clinical trials. In her role as Academic Director of the UQ Clinical Trials Centre, or CTC, she is leading the establishment of the CTC during 2023. And in her other role in STARS Hospital in the Metro North, or the Surgical Treatment and Rehabilitation Service, Professor Foster leads an interdisciplinary team of allied health professionals, nurses, medical doctors, and consumer and community involvement staff across the UQ and STARS Hospital partnership, where they are focused on embedding education and research in the healthcare setting. So please join me in welcoming Professor Foster today.
Thank you very much indeed. I'm just going to share my screen and hopefully you'll be able to see my slides. Okay. So thank you very much indeed for the invitation to come and share where we're up to with these 2 really important initiatives, here at ULTRA and the UQ CTC. So these really are 2 linked initiatives from the University of Queensland. The first focuses on academic capability, and that's the Hero ULTRA team of methodologists. And the second initiative is essentially providing a centralized collaborative research platform that we're calling a clinical trial centre, and taking a one UQ approach to the establishment of that, so that we have here in UQ a clinical trial centre for the future. And hopefully you can see then that the vision is that these linked initiatives will work together to augment our expertise, accelerate our randomised controlled trial capability, capacity, coordination, and competitiveness by bringing together in the visual there different team members with different but complementary areas of expertise, knowledge, and skills to really lift us up in terms of our ability to do top quality randomised controlled trials. Very briefly, why do we need to change? I guess in terms of the randomised controlled trial landscape, my reflection over my career is that these are typically very long roads. They're very long roads from ideation through to close out completion, publication, and impact. And, typically they're 3, 4, or 5 year funded timelines. But then when you add in that ideation and funding application phase, and you add in the publication and impact phase, we're typically talking about long roads of 8 years and more for these large, high quality randomized controlled trials that are really practice and policy changing. And one of the challenges of these long road trials, as I call them, is of course they're relatively inflexible. And I liken them to this, type of large road train or juggernaut that we take a long time to get them on the road. And when they're on the road, we're very busy keeping them on the road and keeping them going to the end of that road. And they're relatively inflexible big beasts, as I call them. And the problem with that, of course, that we're all learning is that if we start just over there on the left, they're very expensive. So these trials in Australia are typically the largest funding envelopes that we ask from funders like NHMRC and MRFF. They're relatively blunt designs in that they are asking typically one key question. They have those long timelines that I mentioned, but we also then struggle to recruit the number that we need. They're typically intervention focused, which means that multiple randomized trials of this type are then needed to evaluate other interventions for particular conditions and diseases. Often by the time the results are known, practice may have actually moved on. Only about 30 to 40% are positive trials in that they demonstrate, for example, that one way of doing things is better than another. And of course, they've been criticised as they take much too long to translate their results into real clinical practice to bring benefits to healthcare, patients, and the population. And so it's clear that innovation in randomised controlled trials is needed. And by innovation, I simply mean disrupting that linear road that I talked about, where we design a trial, we conduct a trial, we analyse a trial, by building in a variety of review and adapt loops. And we can think of that in many ways. And that's what we've tried to do as we've ideated the Ultra team that bring new areas of expertise to the local landscape to improve our ability to innovate in randomized trials. Of course, we've already been busy improving things in this context, and I've listed just some of the areas that we've seen over recent years really changing the landscape for randomized trials, both in design, such as an increased use of external feasibility and pilot trials, or pre-specifying internal pilot trial phases with clear stop and then go criteria, using allocation ratios other than 1-to-1, re-estimating sample size, although, of course, to date, frequently that's because we're struggling to recruit the number we need, as well as other innovations like trials within cohorts and studies within trials. So these things have all been helping us already to innovate in randomized trials. We've also seen a step change in innovation in processes. And of course, we've never seen quite the innovation in processes as during the COVID pandemic. But we've seen steps towards streamlining ethics and other approvals. We've seen a great increase in the use of digital data, including EPROMs. We've seen real shifts towards using different methods of recruitment that speed up recruitment. And a key example, of course, is social media recruitment. And we've seen an explosion of trials that are moving more towards remote types of consent. But it is in particular, I guess, some of the innovations in design that has really underpinned the idea of the Ultra team and bringing new capability, particularly for innovation and trial design. I'll just go back in time to highlight, I guess, how pivotal this particular trial, Stampede, was, which was one of these new trial designs, a multi-arm, multi-stage or MAMS trial and an adaptive platform trial. This trial was in the field of prostate cancer. It started way back in 2005, but essentially what it has gone on to do has been a really great exemplar of how we can begin to do the randomized controlled trial differently. So instead of just comparing a small number of treatments, it was developed to compare many new treatments versus a standard control arm. And then throughout its life course, it has been adding and dropping intervention arms and reporting at different times with different comparisons. It has also changed its control arm because superiority was demonstrated, and so the control was changed to be the more effective standard of care. This is just one example of one of these trials that really is challenging us to do things differently and to increase efficiency. So no stopping and starting of these large juggernauts or road trains, and having many randomized comparisons. And the estimate is that if Stampede had been done in the old way, where we do many two-arm parallel randomized trials, that it would have taken 40 years to get to the results that this trial has already been able to demonstrate thus far. So I guess I'm thinking about where we need to go next in terms of innovation and randomized trials, and that's particularly through design. So bringing in new methods such as refining sample sizes, dropping inferior treatments, oversampling those that are most likely to benefit, stopping the whole trial for success or futility, adding new treatments, and so on. But I'm clear that innovation is not just limited to design. And as part of the Ultra team, what we have aimed to do is add in other roles that can bring innovation in many other aspects. So thinking, for example, about extending analysis beyond single primary outcomes, using better pretrial information, for example, in economic analysis that can better justify the large costs of these trials to funders, ensuring that we embed focused qualitative research that can ensure these difficult to do trials recruit to target, that we work with the challenges of, for example, clinician and patient equipoise, and that we actually use embedded qualitative research to improve the way that we do these trials. Other innovations can be the greater use of health technology, teletrials, and data sharing. And of course, towards the end of these trials, we're really thinking about how we can better build in implementation science, not just as an afterthought, but right from the outset in order to try to reduce time to translation and impact. And we're seeing even innovation, for example, in results and results now being published from trials even before they're peer reviewed. And again, this takes an interdisciplinary team, and that's essentially what Ultra is all about. So on this slide, we've got a team of methodological experts that have been funded by the HERA initiative at UQ. On the left are the three roles that we have already appointed to, and you'll hear today from our Level E Professor in Innovative Trials, Andrew Martin, who has literally just joined the UQ family this week. Secondly, today you'll hear also from Associate Professor Ingrid Hickman, who has been with us now for just over a month in her new role. And we're looking forward to Dr. Natalia Costa joining us in that qualitative research role from January 2024. The team in total represents just under a $10 million investment by UQ. And again, this team is very focused on innovation and providing the academic capability to help us innovate in randomized trials. There's another reason why we need to change, and I take us back to the Ernst & Young scoping study that UQ commissioned in 2021, and a workshop that we held shortly after that. This review really summarised the current challenge in the Queensland and Brisbane landscape, which was that our clinical trial landscape is fragmented, with a range of operators, funding, policies and relationships that all essentially work together to reduce our capacity, capability and interest to undertake investigator-led randomised trials in an efficient manner and to the required standards. And that report highlighted the many lost opportunities, the lower number of randomised trials in Queensland than there should be, the lower number of funding dollars coming into Queensland for randomised trials, and then the particular negative sequelae of that, including fewer patients in those trials, fewer clinicians trained in trials, and the following negative health and wellbeing impacts. And so we've always argued that in parallel to the Ultra initiative, that we should drive forward to action the recommendations that came from the Ernst & Young report. And in purple there is the option that we're running with at the moment, which is to take forward their recommendation to develop a UQ clinical trials unit or centre that is for UQ and UQ partners, which is being ideated essentially as a full-service, collaborative clinical trial support team that can expand to other partnerships as the model matures. Just to be clear, what do we mean by clinical trials centre? So we're talking really about a specialist unit that have the specific remit to design, conduct, analyse and publish these clinical trials, that provides the specialist expert statistical, epidemiological and other methodological advice, and that crucially has the expertise in the central coordination and regulation of these trials to be able to deliver them to time and to target and to the required standards. This is quite different to the sometimes interpreted term of a clinical trials unit that is seen, I think, more locally in Australia as a local clinical trial delivery team or indeed a clinical trial facility. That's not what we're talking about. We're talking about a capability of people who work together to provide an infrastructure for all of the stages from ideation through to close out data archiving and data sharing of these high quality randomized trials. So I'd just like to finish by explaining where we're up to with the establishment of the UQ Clinical Trials Centre and really summarise the processes we're going through of endorsement, engagement and establishment. So in 2022, the UQ senior leadership endorsed the direction to develop a One UQ Clinical Trials Centre and to establish that as one of 11 collaborative research platforms within the university. And that followed a business case submission last year. This year, we've been very busy engaging a variety of stakeholders through workshops and discussions. We've stood up an independent advisory board. Many of the members of that advisory board come from other similar CTC infrastructures across Australia. And we have just recently started the journey of standing up a UQ CTC leadership group. And so, as you can imagine, we're in the thick of discussions about things like collaboration criteria, about transition models for next steps, and about endorsement of what will be our core focus and our core functions. But as I mentioned, UQ CTC will be established as one of 11 CRPs that report upwards in the university to the Deputy Vice-Chancellor for Research and Infrastructure. That means that we're in the process of establishing the wide range of working groups that we will need, that will all be part of this CTC. From a group, for example, that will make decisions about adoption of studies, right through to teams of people who will be working on information and data systems. But by the end of 2023, we'll be submitting the development, our implementation plan with cost estimates to the senior team at the university, and making recommendations about a best fit transition model so that we're ready to welcome UQ researchers and academic title holders to collaborate with UQ CTC as it gets established in 2024. So thank you for listening. My email is on this slide and I look forward to the Q&A after we've heard from Andrew and Ingrid. But if I can, I want to do just a little plug for the upcoming ACTA, so the Australian Clinical Trials Alliance Symposium on International Clinical Trials at the end of November, being held in Melbourne. Last call, I think, for people who want to register to this, but if you're interested in knowing more about the clinical trials landscape here in Australia and want to be part of that journey, do use that QR code, and I'd love to see you in Melbourne. So thank you very much.
Thank you, Professor Foster, for a great introduction to the, the evolving clinical trials landscape, both here in Queensland and in Australia more broadly. Next, I have the pleasure of welcoming Professor Andrew Martin. Andrew is the incoming Professor of Innovative Clinical Trials and the most senior appointment in the University of Queensland Clinical Trials Capability Ultra Program. Previously, Andrew was Professor in the Biostatistics Group at the NHNMRC Clinical Trials Centre at the University of Sydney. He has over 20 years' experience in clinical trials research, gained from senior roles within research-based pharmaceutical organisations and academia. His specific interest is in the application of optimal study designs and analysis methods to the development and evaluation of health interventions across all phases of development. So please join me in welcoming Professor Andrew Martin, who will hopefully be expanding on, I guess, the innovation and design vision for Ultra.
Okay, thanks very much. This is my first day on deck in Brisbane. And on the one hand, it seems a bit daunting to be asked to give a talk so early on. But on the other hand, I'm really excited to be here and actually thrilled to have the opportunity to introduce myself to you. I'm going to talk about some of the topics that Nadine touched upon, but I won't spend a lot of time on them. So some of the issues we have with the conventional approach of clinical trials designs, and talk more about how adaptations to ongoing trials, as well as things called master protocols, can potentially yield great efficiencies in the research that we endeavour to do and the questions we endeavour to answer. And then finally, I'll talk a little bit about my own personal reflections on where to from here. The basic model for the RCT has been around for a long time and was used very successfully from the 1940s to generate high quality evidence on which to base healthcare decisions. That design, along with some of the fundamental features of randomization and double blinding where it's possible, will be coupled with a serial development for new interventions comprising an initial phase where feasibility is established, a second phase where the safety and activity of a treatment is evaluated, and then finally a third phase looking at the effectiveness and potentially cost effectiveness of a treatment to determine whether it should be adopted in practice. Now, these designs were typically fixed in the sense that one would specify all of the elements of the experiment at the start. You would then go out and conduct the experiment, conduct the trial and gather the data. And at the end of that process, you do an analysis and you use the results of that analysis to inform your decision about whether to progress through from phase 1 to 2, 2 to 3. And so what I'm describing here is a scenario where we don't learn from the accumulating evidence from a fixed design until the end of that trial. And that, coupled with the inevitable hiatus that occurs between the movement from phase 1 to phase 2 to phase 3, is a really key contributor to the long duration that it can take to progress a new intervention through this process. So this is really speaking to the lack of efficiency of that model for evaluating, for developing interventions. It can also be unfeasible to apply that kind of approach in an era where what would have been deemed a fairly common disease is increasingly being disaggregated into rarer subtypes of disease on the basis of molecular characterization of the condition. So think, for example, lung cancer is now considered to be a collection of rarer diseases, each with different genetic signatures. So actually gathering enough people with what would now be considered a rarer disease to enroll in the kind of program I described on the previous slide could be very difficult and unfeasible. And finally, this idea of trying to deliver evidence in a timely fashion that remains relevant to clinical practice. There's no point delivering evidence on a question when clinical practice has moved on. And I'll give an example of this scenario a little later. So I hinted at trial adaptations to ongoing trials are potentially one source of improving efficiency. And I'm going to talk about each of these dot points, but not so much the last two that are shaded out. And I want to begin with a really simple example of trial adaptations, and that's adaptations to ongoing trials that actually have a fixed trial design. We know that that's pretty common. We've done an audit of randomized trials registered in the ANZ CTR, and it's really quite common that the reported results and the reported methods differ in some way to what was pre-specified in the protocol. Reassuringly, most of that has been done legitimately. And in fact, this paper, we elucidate the difference, the distinction between what constitutes a legitimate change to an ongoing trial with a fixed design versus one that would be illegitimate and threatens to introduce a bias into the results. That work's actually been cited by international guidelines that came out in response to the impact COVID had on the ongoing trials at play, and provides instruction on how trialists can legitimately go about adapting their trial so that it can remain in operation and it can remain relevant and can react to extenuating circumstances, to unforeseen circumstances, in a way that doesn't introduce a bias. So adaptations to ongoing trials with fixed designs are possible. Nadine mentioned sample size re-estimation, and what that effectively means is revisiting those assumptions that were made when you planned your experiment, your trial initially, and determining whether the evidence that's being accrued are consistent with those original assumptions. So things like the event rate in the control arm will have been estimated. The degree of missingness in relation to a patient-reported outcome measure, the degree of adherence to assigned therapy, the rate of follow-up, and so on and so forth. And these are all examples of inputs to sample size calculations that can be monitored without the need to unblind a blinded study. You don't need to know about treatment allocation for things like pooled event rates, for things like non-adherence to protocol required assessments. And we actually have a good example of sample size reassessment from a trial I'm working on, looking at an anti-inflammatory drug in premature babies, where based on the higher than expected pooled event rate, we were able to considerably reduce the sample size, leading to tremendous efficiencies and getting an answer quicker. We're not there yet, though, however, with that study. Group sequential designs are an example of a type of trial that has baked into it pre-planned adaptations. It has baked into it opportunities to do interim analyses and make decisions about whether there's sufficient evidence to conclude an experimental intervention is effective, harmful, or that continuing the study is a futile exercise in the sense that it's highly unlikely that there will ever be shown a clinically meaningful difference between the experimental treatment and the control. So here's an example of a trial that I am statistical lead on, where we used a group sequential design, and at the initial interim analysis triggered at 50% of the planned number of events, that was deaths in this case, we obtained strong evidence of benefit And very swiftly, we were able to report on that. We got a high impact journal article and a plenary session at ASCO, which is the premier oncology conference globally, attracting more than 30,000 delegates. Now that was a vastly sped up process for getting out evidence and affecting change to guidelines and to practice. It actually took another 3 years for the trial to accrue the total number of planned events. And we published that just recently. Now, another idea for improving efficiency is to try and get rid of that hiatus that can arise moving from phase 2 to phase 3. And one way to do that is to have what's called a seamless phase 2-3 design. Now, phase 2, remember, it's all about looking for activity, and you're going to be measuring activity on some quick marker of future effectiveness. And I'm using an example of the accelerated BEP trial, the phase 3 accelerated BEP trial. Again, I'm statistical lead for this, where we were comparing, and still are comparing, an accelerated chemotherapy dosing regimen against a standard dosing regimen. And in the phase 2 piece of this trial, we're looking at activity as the endpoint. And in this case, that was quantified in terms of favourable response. So something we can measure quite quickly, and we need to see adequate evidence of favourable response to trigger continuing accrual to the study. We had a second test of activity on favourable response. And if we passed that hurdle, we then moved from phase 2 to phase 3. The primary endpoint is now different. It's progression-free survival. And we applied a group sequential design. So we look at whether there's sufficient evidence early on at an interim to declare the accelerated regimen is superior, inferior, or there's a futility to continuing the study. And right now, we're marching forward, having passed those hurdles, through to waiting on 100% of the planned events to be observed. So no gap between phase 2 and phase 3. The study doesn't halt accrual. There's no new protocol amendment or ethics application. It just marches on. Now, some of the other methods to implement adaptations that you see on this slide, I won't talk about, but some of you might recognize these approaches already. Because I want to move on to talk about master protocols as, from a superficial level, a single document that provides the blueprint for undertaking trials to answer multiple questions. And I've got some examples of refinements to that, but they all will share a common feature, and that is that there is a single protocol specifying their features. And once set up, there will be a sort of a single infrastructure, operational infrastructure to recruit patients, to administer treatments, to make assessments, to store data in a single database, to manage that data, and so on and so forth. And in some cases, there are opportunities to share information across individuals that are contributing to different questions within a master protocol. Here's a really simple example of the birth of a master protocol. We had a trial, again, a statistical lead for this phase 3 study, comparing regorafenib to placebo in advanced gastric cancer. It was an international trial. And along the course of enrollment to this trial, the clinical landscape shifted, and this question became of less interest from a clinical perspective, placebo being no longer considered a reasonable, or placebo plus best supportive care, I should add, no longer being considered an optimal control in this group of patients. So we put that study, let's just put that study to one side for a moment and think about, well, what would a contemporary study answering an important question look like? And we came up with the figure on the right-hand side of my screen and of your screen, comparing a combination of regorafenib and nivolumab against not placebo, but investigator choice chemotherapy. Now, what we did is we didn't just terminate the first trial, we rebadged it. It became Integrate 2A. The new trial became Integrate 2B. And rather than seeking approval for a whole new trial, we grew the original protocol into a master protocol. So it's a single protocol that specified all of the instructions for undertaking both of these evaluations. So in practice, it meant that there was no gap between the finishing off of Integrate 2A and the commencement of 2B. The way we did it was we put through the protocol amendment through ethics, and as centres were brought on board Integrate 2B, they were closed out from Integrate 2A. So this kind of overlapping period is the period where that process was occurring. So we didn't lose any momentum, right? So a nice example of a simple way of how a master protocol came about. Nadine's already mentioned the Stampede trial as an example of a multi-arm, multi-stage trial. I'm not going to talk much about it. Suffice to say that you can see from the start, it was planned to have multiple arms and a single master protocol describing how these arms should be administered and how comparisons should be made. Each comparison, by the way, is being made against a single common comparator. Now, what about the multi-stage part? Well, that's harking back to what I spoke of in relation to group sequential designs, where you have interim analyses baked in, and if you're getting insufficient evidence to warrant continuing recruitment or follow-up to an arm, you prune it away. So you can see some of these arms are shorter than others. And another really exciting feature for that Stampede demonstrates is once you've got the infrastructure set up to run a platform trial, a multi-arm, multi-stage platform trial, you can actually not only cut away or prune away ineffective experimental arms, but start to add in new arms as external data shows promise for those interventions. If you're going to give a talk on innovative trial design, you kind of have to mention umbrella trials and basket trials, but the names are super confusing and no one can really ever appropriately explain why one is called a basket trial and one is called an umbrella trial. And if you ask two different people, they'll give two different answers. But some of this has evolved from what I mentioned earlier about improvements in fundamental biology and assays, allowing us to make finer and finer distinctions between the characteristics of diseases at the genetic level or other omic level. So an umbrella trial would involve one particular disease being disaggregated on the basis of some marker or other. It might be a genetic marker, or it could be on the back of some demographic characteristic like frailty or age, might be a better example. Frailty would be more a clinical characteristic. And those subsets within that disease undergoing exposure to a particular intervention that is seen to be most relevant for that subset. And you could do that as a series of single-arm studies, or these could be randomized comparisons. With a basket trial, it's a little bit different. You've got different types of disease that all share some feature that a treatment is going to target. So in this case, we've got different types of cancer that all share some common genetic mutation that a particular targeted drug affects. So the MOST program is an example of a trial run at the centre I've come from, where individuals with cancer of any type and no further standard treatments available undergo a molecular screen and get funneled to a particular arm that will expose them to a therapy that is hoped to be most well tailored for their particular molecular signature. So you could sort of think of this as a type of umbrella trial, whereas the Paragon trial is taking women with gynecological cancers that have a particular characteristic in terms of ER and PR positivity, and the type of cancer will vary, but they're all undergoing the same experimental treatment. So a type of a basket trial. So I'm gonna wrap up, and I've talked a lot about this dot point, efficiency via adaptive and platform trial designs. That's kind of my area, but I want to throw a few extra things out there, and Ingrid's going to talk more about some of these as well. But I want to throw out there the importance of having formal ways of prioritizing what constitutes an important question and where it makes sense to invest in trials research. Having end user involvement in developing trial plans is super critical, having reviewed many grant applications and being aware of the importance that funders place on that, super important. Having evaluation regarding feasibility through qualitative and other embedded mechanisms is really important to make sure that issues are identified early on in the trials process. Having a rigorous implementation plan that means that the evidence generated is not left just gathering virtual dust in a virtual journal on a virtual shelf in a virtual library, but actually being used to improve health outcomes, completely critical. And one of the things that I'm really interested in these days is doing more to try and embed trials research, randomized research into routine care, making use of available mechanisms or routine mechanisms for collecting outcome data. So look, thank you very much. I'm going to hand back to our chair and we'll take questions a little later.
Thank you, Professor Martin. That was a very interesting and enlightening talk. I guess complementing Professor Martin's talk, we now have Associate Professor Ingrid Hickman. Associate Professor Hickman is an implementation scientist with more than 20 years experience in health services and clinical research. Her career has focused on research excellence, strategic leadership, and translating scientific evidence into improved clinical care for people with complex chronic conditions. From randomized control trials and mechanisms of disease progression through to patient-centered co-design of health services, her collaborative approach to research aims to find solutions to healthcare problems. So please join me in welcoming Professor Hickman.
Thank you so much. I was asked to introduce myself, and I think a lot of people may find my face familiar. I have spent 20 years on the PA Hospital campus, participating and leading clinical research trials as the lead of the Metabolic Obesity Research Group. So that's been located here at PA, affiliated with UQ, and of course here at the lovely TRI. Andrew and Nadine have spent time in their talks talking about the need to innovate clinical trials. I've learned a lot over the time that I've spent working in trials and know how critical that they are to progress and change the way that we do practice. But we also know that we are faced with a number of problems, and this leaky research pipeline where there are a number of stages where we can find inefficiencies are what Ultra is all about, to try and plug those leaky gaps. And implementation science can play a significant role in that. So how do we get what works to the people that need it and with a greater speed and fidelity and efficiency? Well, before I talk about implementation science, I wanted to just give a palette cleanser from all of those study designs that Andrew's described. And also for Andrew's benefit, I've chosen an analogy for a Queensland summer that he's about to suffer through. We've got 3 children jumping into a pool here. Can someone call out to me, what are these children wearing? Togs. What was the other one? You're all Queenslanders. Swimmers, bathers, boardies. There's many, many words you can use to describe pretty much the same thing. And implementation science has in some way suffered from that because across the breadth of the science, many different words are used to describe the same thing or similar things. And in fact, you may find that you yourself have been already doing some implementation science in your long careers in science, but may not have called it implementation science. So what does implementation science mean to you? It's okay if it doesn't mean much as that terminology, but what the implementation scientists define it as is the scientific study of methods to promote the systematic uptake of research findings and other evidence-based practices into routine practice, and hence to improve the quality and effectiveness of health services. That's a pretty academic definition, if you ask me. And when I like to talk about implementation science, I do like to use some very non-scientific language to put it into context. So if you think about what gets you up out of bed every morning to do your science, what you're creating in your research world, the intervention or the practice or the innovation, let's call that the thing that you're interested in. And effectiveness research looks at whether that thing works. So that's your randomized controlled trials, those innovative designs that help you understand if it's going to be effective. So implementation research looks at how best to help people and places do that thing. And implementation strategies are the stuff that we do to try and help people and places do the thing. So when we're measuring our main implementation outcomes, they are around measuring how much and how well are we doing the thing. And so potentially in your experience, you've been doing a bit of this and calling it other things. And potentially you've been using implementation or thinking of implementation as a bit of a clip-on or an add-on to the work that you're doing. You may have been writing a funding grant and there's a box you have to fill in about how am I demonstrating research impact? How am I engaging with consumers? Demonstrate how you've shown end user input into the design of your study. And so I think for many of us, that is what it's been in the past. And so I guess where Ultra is coming to this is that we would like to try and change that approach and ensure that the shiny black frames of the glasses that are created by the statisticians don't just have the implementation as the rosy sunglasses clipped on top, but in fact are embedded within the trial design from the very beginning of the pipeline. These can improve efficiencies by having implementation science embedded early on so that you have measured robust data throughout the course of the trial to help you understand the why of why a study might or might not have been successful. And so traditionally, I think the research pipeline starts with efficacy studies that then lead to the effectiveness studies, which then are often, once that answer is known, may be handed over to a completely different group of people to do the implementation work. And that is often how it works in health services. And what has been proposed is to start to think about hybrid designs or innovating designs to bring that implementation research expertise into the effectiveness studies and design what is called a hybrid design or hybrid study, which combines the elements of both clinical effectiveness and implementation research. And so there's been a number of different types of hybrid implementation effectiveness studies proposed. Type 1 hybrid studies is where there's still a lot to learn about the effectiveness of an intervention or of a thing. And so you're testing the clinical intervention as your primary aim, the effectiveness of that intervention. But you might, during the course of that trial, also collect information on the context of the implementation. You might collect information on barriers and enablers to whether you'd be able to implement this if it's shown to work. You may interview patients to ask about their perceptions of the treatment, their experience of receiving the treatment. There's a hybrid type 2 designs, which are probably the more complicated, complex designs where you're simultaneously testing the clinical intervention as well as the implementation intervention or strategy that you've chosen. So in essence, you have two primary aims. You have the effectiveness primary aim. Is it effective, this clinical intervention? But you're also looking at feasibility and the potential impact of this intervention in a clinical setting. So outcome measures in that sense might be around measuring adoption or fidelity within the real world clinical setting, or factors related to uptake in the unit or sustainability over time. And the third hybrid design is where you may be really confident in the effectiveness of your thing. So there's been many systematic review and meta-analysis to say that this thing works. We know it works, but we don't know how to deliver it or how to implement it in practice. So the primary aim will be designed around testing implementation strategies, but then you may also add in clinical outcome measures to that. So potentially as a pre-post data collection across the different implementation strategies, so that you can assess whether the clinical outcomes achieved within the context of the implementation are acceptable or adequate. So there are examples of 3 hybrid designs where you can start to embed implementation science within your randomized controlled trials. So implementation theories and frameworks really are a way for us to contextualize and build on our knowledge of what works, where it works, and why it works or doesn't work. And there are many, many frameworks out there to use. So a recent review has identified over 150 different frameworks to choose from. So there's no one size fits all. It's all very contextualized based on what you need and your setting needs. But Ultra is really designed to help to demystify this and to help to embed the most appropriate implementation frameworks into clinical trials. So we will bring the methodological expertise in implementation, as well as statistics and health economics and qualitative research to really add value to teams who are undertaking clinical trials. That's me, thanks.
So thank you, Associate Professor Hickman. And now I have the pleasure of welcoming all our speakers, including Nadine, who will be joining us on Zoom, up to the stage for a Q&A. So the audience, whether you're on Zoom or out in the auditorium, please prepare your questions. So do we have any questions from the floor? We have 2 roaming microphones, so... Right, well, I guess I'll get us started with a quick question. I guess, Nadine, you can perhaps give us a more Queensland perspective, whilst Andrew, you can probably give us a more broader overview. But what do you think is the biggest hurdle, I guess, for researchers taking this step to translating into clinical trial, particularly those working in more the bench sort of laboratory side of things? It does feel like sometimes there's quite a big chasm between there and a lot of inertia, I guess, between identifying potentially a pathway or a compound and actually bring it to clinical trial. So I guess we could start with Andrew, you first.
Sure. Well, look, that's a big question. The challenges of taking an intervention from the bench into trials. I think that finding key collaborators is absolutely critical, and finding experienced investigators to partner with is absolutely essential. And to help facilitate that process, it's really critical that someone with an idea is able to articulate that idea very succinctly. And we talk about a one-page concept outline, something that a person should be able to read and kind of get what you're interested in doing. And you should also have your elevator pitch ready. Okay, so that's how you describe your vision for a trial or your vision for evaluating an intervention in the time it takes for the lift that you're sharing with a senior colleague to go from the second floor to the fourth floor. Right? So look, if there was one thing I would prioritize to answer your question, it would be that. And there's one or two other things, but I won't hog the floor.
Thank you for that. Ingrid, do you have any follow-up?
Sure. I mean, I think implementation frameworks can be really helpful to to allow you to see the context of the setting of where you're doing your research. So who are you doing the research for? Do you have, what's your impact statement? I guess that's similar to what you were saying in terms of, do you have a clear understanding of the purpose of your research, and can you work backwards in understanding all of the key stakeholders who are involved in order to get from your science to that impact? And so then doing some stakeholder mapping about who is involved in that moving beast of moving science from the bench to the bedside, and what role can you play in helping to develop and establish those relationships with those stakeholders.
Great. And I mean, Nadine, you showed some pretty, I guess, stark statistics about why Queensland is perhaps not leading the way in clinical trials uptake, and maybe you have some specific insights to why Queensland we're perhaps lagging behind.
Oh, I would say in brief, just because I know we're short for time, I think Health Translation Queensland have led on a really useful report that helps us understand some of those challenges, but they include lower numbers of applications going in from Queensland. So that's partly what we're here to try to help with in terms of the Ultra team and UQCTC in future. But also we need more clinician scientists. And those of you who know me will know I'm also a passionate advocate for clinical academic careers. And we need to be growing the next generation of people who walk both careers. They're both clinicians and they are scientists and researchers and trialists. And so my hope is that over time we'll be able to help with that by providing training and mentoring opportunities to expand our clinical academic workforce here in Queensland.
Thanks. Oh, we've got a question over here. We'll just get a microphone to you so that they can hear from Zoom on Zoom.
Thanks, great presentations. I understand that there's a number of existing clinical trial units in already within UQ with collaborations around Australia. I was just wondering how would you go about navigating how will it work with other existing trial units?
So I'll take that one if I may. I mean, obviously what we're trying to do is move forward to essentially offer a one UQ approach to a clinical trial centre. So our current landscape is fragmented, and we have some great pockets of excellence in clinical trials at UQ. And what we're hoping to be able to achieve is to harness their expertise and to ensure that as we go forward, we share expertise so that that becomes more available more broadly to others. So it is about changing our approach from our current fragmented model to, in the future, a more coherent one UQ approach. We're currently discussing different transition models, and no decisions have been made on that yet. But clearly, the vision is that we have a one strong infrastructure and interdisciplinary team going forward who are able to collaborate on trials from ideation to closeout, irrespective of the condition or disease in focus.
Thank you. I'll just pass quickly to Amelia, who has a couple of questions on Zoom, and then we'll try and pass back to you, Ranjini.
So we've got two questions which are quite similar. One of them is this, okay, sorry. They wanted to know whether the HERA in Ultra is free for UQ researchers. Is it a pay or per service? And what is the decision-making on the process about which projects to contribute to if the workload is too high? And if it's, and which projects to fund? Yeah.
Let's take this one.
Well, I think that might be one for Nadine. And the question is really asking how are the expertise sets at Ultra and the future clinical trials centre going to be accessible to others, and how are things prioritised and what will the costs be? I mean, these aren't really questions I'm in a position to answer, but certainly there's a great focus on supporting University of Queensland initiatives and ideas, and simultaneously helping to support and grow the research across a more broader range of organisations and institutes. And with that very general remark, I'll hand over to Nadine.
Yeah, and look, I guess what I would say is the CTC is already developing some collaboration criteria that once finalised will guide the decisions of a CTC adoption group so that we make transparent and hopefully wise decisions about which studies to collaborate on. And I keep saying we have to walk before we can run, so we also have to build the CTC in a way that will be risk appropriate and sustainable for the future. So our aim is to be able to have collaboration criteria that will then be on websites and become transparently known. In terms of Ultra, well, clearly I think you can see that what we're trying to do is attract a team of people who have additional expertise for some of the innovation that we need. Not all trials will need the level of expertise that folks like Andrew and Ingrid bring. And obviously what we need to do is ensure that other mechanisms of support for taking, for example, your first step into a randomized trial or building a simple 2-arm parallel or 3-arm parallel trial, there are already folks within our landscape who will be able to bring expertise and support and services that already exist to help with that. So whilst all of this is yet to be determined, now that Andrew is started and Ingrid has started, inevitably the questions I would ask is really centred on, does this issue, does this question, does this potential randomised trial need the expert support of a team like Ultra? So we hope to be able to simultaneously work across the CTC and the Ultra team so that we can spot those questions and studies as they're in development and wrap around the support of Ultra into those particular studies that need that particular expertise.
Thank you.
I hope that answers your question, but very happy to receive further questions on email and contacts and discussions as we go.
So one more, yeah. So we have another question. So to perform adaptive clinical trials are relatively new in the RCT space. So if you propose one for a category 1 grant, do reviewers have enough skills to score the grant?
So Andrea, I could perhaps just mention that, of course, whilst Queensland might be a little bit behind some of the other states in developing and getting up and running some of these new trial designs, there is growing expertise across Australia. And my understanding, particularly for category 1 grants, is that some of the statisticians that work within subgroups like Ostrom and methodological subgroups, I'm told by them that many of them do sit on these category 1 funding panels, albeit they may be the only person on the funding panel with that kind of expertise. I'd also just like to flag that within the umbrella of ACTA, there's a special interest group in innovative trial designs, and they are offering a rolling programme of online opportunities to present on these new trial designs and receive really helpful feedback. And that has just started this year. So if you're thinking about designing one of these, there are even national groups now who are willing and providing expert support in the ideation phase for these new trial designs. So I'm really pleased to see that develop. And again, if you want connected into that, then drop me an email and I'll connect you in. Andrew.
Yeah. If I heard correctly, it was how do you access expertise for these trials and how do you get funding for these trials? Is that about right? Well, I think I would support what Nadine has said, and I'm very keen on growing the collaborative network that is in place with AusTREM, which is a group of senior biostatistical academics collaborating in innovative and adaptive trials research, and in growing that network and strengthen ties to UQ. In fact, there's an AusTREM conference going on at the moment, and I was down in Melbourne talking to a number of investigators of the AusTREM collaboration yesterday, and they are very enthusiastic to bring UQ and Queensland more into the fabric of the group. I personally want to help grow expertise in this area amongst current UQ and Queensland personnel, and I'm interested in having AusTREM be an important part of that process. So we have, rather than pockets of expertise, we've got a national framework with strong international ties to leaders in this kind of methodological area in Cambridge, for example. With regards to funding, I think everyone gets a bit nervous about putting forward a grant proposal that has a design that's pretty tricky to explain. If you've sat on a panel before, you'll know that you've got to review a lot of material. In the panel discussions, you won't get a lot of time to talk about it. And frankly, the more complicated the design that's being proposed, the more confused people will be on the panel, and the more difficult it will be for the primary spokesperson to really champion what they might perceive as being a great trial. So I think that is fundamentally difficult, and three things need to be borne in mind. One is complexity should only be introduced in a proposal when it makes sense. So just turning something that could be undertaken as a simple 2 or 3 arm RCT, turning it into something more complicated because it's perceived as being in vogue at the moment, is probably a bad idea. and reviewers can sort of sense that. The second is to try and not only explain the rationale for the more complicated design, but explain it in as simpler terms as you can, and perhaps having reference to internal documents or reports that can be accessed through websites and so on, that can provide a little bit more elaboration on what you're planning to do. Now, having something published would be ideal, you know, a protocol paper, for example, but the time taken to get that through is too long, I think. So I'm quite keen on the approach that I outlined there. And it could include, for example, detailed output on simulation studies showing the operating characteristics of the design you're putting forward. And finally, the third thing was getting investigators, collaborators with expertise in the area. That's absolutely critical. So once again, reviewers will say, well, it sounds good. I'm not exactly sure of the complicated design, but I can see blogs is on the list of investigators, and blogs is has quite an impressive track record on this area, and they'll take comfort from that. So they would be my three sort of tips in relation to grant application strategies.
One of the additions just is that I think we're leaning into a bit of learning from the UK here in Australia. So there are discussions about the need for a funding stream for essentially the early phase development of these types of new trial designs. So the NIHR has stood up such a funding stream in the UK, and that's proving rather successful, where they offer a small amount of funding for 1 or 2 years to essentially do the development work of, for example, these new adaptive platform trials, because they understand that they require much more in terms of planning, simulation, master protocol development, collaboration, and so on. So with any luck, I would say that it would be nice to see the funders here picking up on that. And I know that those discussions have been being had here in Australia, taking the learning from the UK, that is a particularly helpful way to get early funding for these more complex randomized trials.
I am aware we're eating into lunchtime. Are there any more questions from the floor before we move on? One quick one over here.
Thank you. They were great presentations. I'm just thinking from an ethics practicality point of view, have you found it useful to present or educate to ethics committees about new innovative trial styles? And I guess it would be great to highlight to them, coming from an ethics committee, what the difference is between a master protocol and just another version of version 1, I guess.
It sounds like a great idea for a publication, frankly. Mm-hmm. And then you could cite that publication in your ethics application. I mean, at the Austrim meeting I was at yesterday, there were folks who sit on an ethics committee there, and they'd kind of taken the initiative to find out more about adaptive designs. So yesterday was largely a workshop in adaptive designs, and I thought that was fantastic. I gave that example of the Integrate program of research, where we grew a protocol into a master protocol. And just between us and whoever's watching this on YouTube, I had some skepticism about the committee endorsing that proposal, but they did. And I told myself, things are changing and people are becoming more aware of the benefits and the merit of these more efficient designs. And, you know, ethics, I mean, I sat on a clinical trials subcommittee of an ethics committee, and the breadth of protocols that came our way was vast and varied. And some of them are just outstanding. And I would think to myself, ooh, what are they doing here? I better read up on this. And I would learn, and then I would apply some of the statistical techniques that were being deployed. And then others were, frankly, you know, not very good. So ethics committees, they have the opportunity to really be exposed to some wonderful proposals as well as some less wonderful proposals. And so I think a publication like the one I jokingly proposed would be a really good idea. Perhaps one already exists.
Yes.
And it does? Oh, good.
Well, I think Roberta's reminding us in the chat that there is an active special interest group that have taken on this task to try to develop information and educational material for a wide range of audiences, consumers, patients, but also HREC or ethics committees. So that work is ongoing as part of the ACTA special interest group.
Yeah, okay, terrific. So I think, you know, gold standard adaptive platform trial proposals will come through ethics committees. They'll increasingly be able to discern what represents a solid, scientifically sound proposal from what is something that's been whipped up and perhaps overly and unnecessarily complicated. Yeah.
Ingrid, I just thought before we wrap up, maybe you can comment. I imagine a lot of these issues which apply to, I guess, workforce upskilling and ensuring the broader workforce associated with clinical trials applies as well to the integration of implementation science into your clinical trials. So I don't know if you have any kind of
Did
final
someone
comments?
from our trip plant that question to you? So as a side hustle in my other job previously, it was around developing teaching and training for the clinical workforce to use the research in their everyday practice, so translating research into practice. And that in itself is a highly specialised skill to train the clinical workforce to use research and apply it, and is often overlooked. And so it's often assumed that once the research is available, that will be equal to being accessible, which will then equal to change in practice. And of course, that's not how it works. And so including that investment in the end user, whether that be the patient or the clinical workforce that will need to use the research, incorporating them from early stages, but also thinking about how the workforce needs to be trained in order to uptake and adopt and importantly, sustain the implementation of that research is really very critical. Implementation science has many frameworks that we can use to help us think about what are the needs of not only the individuals within the workforce, but the settings and the environments in which they work.
Thank you. Well, in the interest of time, we might wrap up the Q&A session, but it's been a really fascinating session. I can see this is a rapidly evolving landscape, and I will pass over to Amila to close today's session.
Okay, cool. Thank you, everyone. Thank you for our speakers, for everyone in audience and our organisers for attending this lovely, wonderful presentation and series. For those of you in audience, please join us in the atrium for a networking lunch. Thank you.